Hey everybody, and welcome back to Blackboard Biology. So in this video, we're still working on protein synthesis, which again involves RNA, but this time we're going to look at the second big section of it, known as translation. So to kind of refresh a little bit and recap from the last video, we have this idea of the central dogma of molecular biology that DNA will be used to code for RNA, and then RNA will then be used to make polypeptides, or later those will get folded and be complete proteins. So this is the central dogma, the central idea for molecular biology is that we have DNA, RNA, and polypeptides. So what we did in the last video was we looked at this idea of DNA to RNA. Well, in this video, we're going to look at the second part, mRNA to a polypeptide. And so this idea right here is known as, this is a typo up here. This should actually say at the top, translation, not transcription. I must have missed a slide. Translation. All right, translation. And in that process of translation, how in the world are we going to get from mRNA to a polypeptide? Well, we're actually going to use all three types of mRNA that are available to us here that we mentioned in the previous video. We have messenger RNA that's carrying the code from DNA to the ribosome. We have tRNA, which is going to be carrying the amino acids that we need to build the polypeptide. And then we also have ribosomal RNA that's going to come into play because it is components of the ribosome that is going to actually put the polypeptide together. And so we're just going to jump right in. It's going to start off here with the messenger RNA. So here is our mRNA strand that we got through transcription, and over here we have a ribosome. Ribosomes are produced in the nucleus in a special region known as the nucleolus, and again, largely they have a lot of ribosomal RNA, so that's part of the reason they're made in the nucleus, because you need DNA in order to make that RNA, and they're also made of protein components. You'll notice it has two subunits, kind of this top larger one and the bottom smaller one, and what it will do is these two subunits, I have already drawn them connected together. It's not technically how they should be. They're actually separate. They will come and join together and attach to this messenger RNA, as it looks kind of here. So you can see here the ribosome has attached to the messenger RNA, and the messenger RNA, you'll notice somewhat, I didn't draw it perfectly, these three bases right here line up into this spot right here, this purple spot. Okay, this is, there are a few different sites in the ribosome where the messenger RNA will align with it, and what we have over here is tRNA that will also align at those sites. But let's get into the specifics a little bit more of how exactly that will work. So I drew attention here to these three bases that line up right here. Well, in the DNA RNA language, three letters makes what's called a codon. Okay, so let me kind of erase this arrow here. All right, so a codon is a group of three. That's on the mRNA. And each codon is basically like a word in the DNA language. Okay, so each word is three letters in the DNA language, and they are going to code for a very specific amino acid, which I have drawn here and labeled as number 1. So this is kind of all like paperwork here almost. The messenger RNA is the specific things, hey, we're going to attach these things in this specific order. The tRNA is kind of like a truck that's transporting the specific amino acid. And if the mRNA has a codon, well, the tRNA has a complementary anticodon. And so that anticodon has to match up with the mRNA codon. And in this case, it does. We look here and we see, I may have to switch to a different color so we can clearly see it. Over here we have a U, A, G, and here we have the complementary A, U, C. Okay, so they're complementary, and what will happen is that tRNA molecule carrying a specific amino acid that I've labeled number 1 or version 1 or amino acid 1. They actually have names. There are 20 different amino acids that are found naturally in life on Earth, and they all have different names, and each tRNA only carries a specific amino acid. And so it is going to slide into this first slot like so. So now it's slid in, and what's going to occur is we're going to see, does the anticodon match the codon? All right, and in this case, big check mark, yes, it does. So the beginning of our polypeptide chain has occurred here. We have our first amino acid in the chain. Well, you'll notice there's a second site here right next to it, and what is going to occur is that another tRNA molecule will slide into that slot. And if a match occurs, which it does, A with U, G with C, A with U, great. Big O check mark, they match, and what is going to occur is the ribosome will take this amino acid from the first tRNA and attach it to the amino acid from the second tRNA. I have labeled this second amino acid 2. Okay, so it's amino acid 2. And what will then happen is now that it has slid over and it has attached, we have the beginnings of our polypeptide chain. So this tRNA molecule right here is now, it's dropped off its cargo. It will leave, and it will actually go back out to the cytoplasm. It can pick up another of the type of amino acid that it carries. In this case, it'll go back and pick up a 1 amino acid because I labeled that amino acid 1. Again, these have real names. I tried to just make this video very simplistic. So perhaps, technically speaking, which I'll explain here in a moment, this all begins at a start codon. Every mRNA molecule has a start codon where all of this is going to begin. Perhaps it's the start codes for methionine. That's an example of an amino acid. We would have abbreviated that MET, and there'd been a methionine. But instead, I just chose to use numbers for the amino acids, okay? So it will leave, and the entire sequence, so it's out now, it's left, it's slid out. And so what are we going to do? How are we've already read this. How are we going to get to the rest of it over here? Well, what we're going to do is this entire thing is going to shift this way. The ribosome is going to slide the mRNA down. This tRNA will slide over into the first slot, and the polypeptide chain will move with it, like so. Okay, so again, this was our first tRNA that matched up here, okay? It has slid out of the way. The messenger RNA has slid over. We're reading the messenger RNA 5' to 3', by the way. This was our second tRNA. Now we have a third tRNA that's going to slide into the second slot. And you'll notice that it actually has the same anticodon as the first one, so it's actually carrying the same type of amino acid 1. So our sequence of amino acids that we're building is actually going to be 1, 2, 1 so far. It'll slide in, all right? And again, this growing chain is going to be slid over by the ribosome and attached to the new one, like so. This tRNA that's now empty will get kicked out. It will go pick up a new amino acid eventually. I'm not drawing that here. And again, this whole thing is going to shift and slide over. Okay, here we go. I'm running into a little bit of technical difficulty. My program has chosen to stop responding for just a moment. Let's see if it unfreezes. All right, so here we go. We've slid over everything, and now this growing chain will attach to the new amino acid there, and we'll get a growing polypeptide chain. And what we see here in this diagram is we've actually run out of messenger RNA at this point. And so what I would like to explain is, again, this would go out. And what I would like to explain is that in real life, mRNA has a start codon. It's a specific sequence of three bases that says to the ribosome, hey, start here. This is where you want to attach and begin things. And it also has a stop codon at the end that says, hey, it's time to release everything at this point. I did not really draw those in. There would have been a start codon up here at the 5' end, and there would have been a stop codon here at the end. There is no tRNA that would match the stop codon. It would be a kind of a protein look-alike that would be somewhat similar in shape to tRNA, but it's a protein that's actually a releasing factor that when it comes in, when there's a stop codon, it actually triggers everything to be released. The messenger RNA is going to leave. It can actually go to another ribosome and be reused. These tRNA molecules leave. They're going to go pick up, again, specific amino acids, but they can go pick up new amino acids. The ribosome will actually break apart. These subunits will actually come apart again. And up here, we have our beautiful polypeptide. Now, in real life, a polypeptide is not going to be only 4 amino acids long. I simply wanted to make a simple video. So in actuality, it would be much, much larger. You would have a much, much longer polypeptide chain. And we're talking hundreds of amino acids long. You know, very, very long. They can be extremely long in length, polypeptides can. And those polypeptides then would go and they would be, many of them will enter into the rough ER where they'll be folded into particular shapes based upon their amino acid sequence and some side chain interactions. And they'll perhaps be attached with other protein subunits to make a complete protein. And so I hope that this video has been helpful to you in understanding translation, that we're going to begin with mRNA. It's read by ribosomes. It has codons. The tRNA has anticodons that must be complementary and match, and we're going to make a polypeptide chain. As always, I want to encourage you, like my videos, comment, give me requests for new videos you'd like to see, and subscribe. And as always, thanks for watching.